really excited to present on what is a very new project for me as well. I want to begin by thanking my co-authors, particularly Alex Ponifrak, the PI on this project, and Chris Carr, and then Hannah Dion-Kirshner is a former undergrad of mine who has done most of this work in the lab, so very grateful to her as well. And then our funding sources, this is a recent NASA exobiology funded program. Okay, so it shouldn't be surprising to any of us that salt is a good preservative, right? So humans have been preserving things in salt for a very long time, and so there's an intuition here that this might work. But understanding how this process works is really important for astrobiology because it's going to affect the biosignatures that we see. We saw some really beautiful work from Lou yesterday showing these relict biosignatures and just like, poor little eukaryotic shells hanging out, but they still have lipids, they still have biosignatures, so we need to think about how these processes work. And so key questions that we're asking on this project are, do different salts preserve things differently? We're working in magnesium sulfate brines. And do different organic compounds preserve differently? So are lipids different than DNA, are different from RNA, and so on? Alex is going to tell you all about why those things should be true and how that works. I know very little about that. And so the question I'll be asking is, how is organic matter incorporated into sediments and salts in this kind of hypersaline basin? Okay, so the sites. We are headed to over here in the middle of fabulous nowhere, British Columbia. Really the middle of nowhere, guys. It's great. And so zooming into these lake sites, there are 3 of them, and look at that. So the farthest south is Basque Lake number 2. This is a site that has a variety of previous work on it, just a little bit. And you can see the really beautiful, so this is a little light in this view, but you can see the spotted character of these lakes. So it's a basin that have sub-brine pools that make them really interesting to look at, and means you can run around on this and sample a bunch of different compositions all at the same time. So that's pretty neat. So that's one site. And then we have Clinton Lake, or Salt Lake, in the thriving metropolis of Clinton, British Columbia. It is sometimes a salt pan and sometimes more of a lake, so you can see it there. And then the optimistically named Last Chance Lake, which is up here on the far north. This one is interesting because it's a chemical comparison, and so this one has some bicarbonate and some sodium in addition to the magnesium and sulfate, which Alex will tell you all about, whereas these ones are much more strict magnesium sulfate brines. Okay, so what are we doing? So the goal here is to figure out what's being produced in these systems and how that's being translated into various sedimentary archives. And so you can go to a lake, collect some water, in my case, filter that water down onto glass fiber filters to collect organic matter that is being produced in situ. Then also go into the more solid reservoirs. So this is some green salt for you. So these are organic compounds being incorporated into salts. Then also the sediments. Then collecting modern biomass. So this is a beaker of slime collected by Chris Carr. So this is a big microbial mat. And then also things like macrobiology. So these little wee beasties swimming around. Because we need to know what sorts of organics are being produced in these systems and how they're getting incorporated. So then, this was the summer, and I went along on this trip, and it's fabulous. Then we went in the winter, and by we, I mean I sent my poor undergrad to go sample when the lakes looked a little bit more like this. I had to teach, and also I'm weak. And so in this case, you got to drill down through that ice to then tap into both the ice itself, which we saw some posters on, and you can melt that ice, filter it down onto a filter. You can get the sub-ice brine, which I'll be referring to as brine as opposed to water. Filter those down, and then also get down deep with a ladle and get some sediment out from beneath in addition to the salts. Okay, so then what are we going to do? So everything in our little organic jars is the sample, and so we're going to first freeze dry and homogenize that sample, and then analyze a variety of bulk parameters. And so I'm interested in carbon and carbon isotopes and organics, and so you need to first characterize the inorganic baseline of those samples. And so we're looking at things like dissolved inorganic concentrations and isotopes of carbonate. Then decarbonate and focus on that organic matter. So how much organic carbon is in there, total organic carbon and total organic nitrogen, and then what does the isotopic composition of those phases look like? And then the real lab work fun begins, and again, bring in the undergrads. And so this is where we spend a lot of time doing lipid extractions via super time-intensive Bly-Dyer approach, which preserves intact polar lipids, and so that allows us to look at those complete lipids together. And then also you can hydrolyze these things, separate them, clean them up, and identify compound-specific biomarker abundances, and hopefully eventually isotopes. Okay, so first step, look at the waters. And so if we look at these things under the microscope with just a little bit of crystal violet stain, you can see beautiful, beautiful cells. This is a 10 micron scale bar, and so these are actually algae, so that's what's making these bright yellow, but you can see abundant cells. We don't have cell counts yet, but these are all the same amount of fluid just in a little reservoir. If we look at the sub-ice brines, they look almost exactly the same. And so I was surprised going in, you still have those algal morphologies, you still have nice round cells all over the place. So, how much organic matter is there? And so if we look at the total organic carbon, you should maybe not be surprised that microbial mats are full of organic carbon, but these sediments are also quite rich in organic carbon. If we look at the total organic nitrogen, you can see that that's mirroring the organic carbon really well. It has like a R squared of 0.97 and a slope of 10 to 1. So, well correlated. And so what do the isotopes of those look like? So this is a little bit more interesting, and here I've plotted these by lake, so the Basque Lakes, Last Chance Lake, and Salt Lake, and then they're colored by what type of sample that was. And so you can see some differences between the Basque Lakes. They're relatively enriched compared to the Last Chance Lake or Salt Lake. You can also, if you look at the colors of these, you can see that the salts are enriched relative to the sediments are enriched relative to the sub-ice brines, really clearly there. So that's an interesting trend. So we need to look at our inorganic phases to see how these compare, because that's setting your baseline. And so if we look at the isotopes of DIC and of carbonate, you can see that this does explain a little bit of these trends. So in our DIC, for instance, this one is quite enriched, whereas this one is quite depleted. So you've got about a 5 per mil spread there, but this one isn't. So this is not explaining all of our data, which suggests that you might have some distinct biological fractionations changing the organic isotopes within that system. If we look at the nitrogen isotopes, there are also some kind of interesting trends. For those of you that work in the system, so the 0 to 15, that's a really enriched nitrogen isotopic value. We're sitting here kind of around 10. Quite enriched, just fixing in 2 out of the air, you'd get 0 or 2. And so these are super enriched, suggesting processing of nitrogen in the system. Usually denitrification gets blamed on enrichments like this. Also, though, in alkaline lakes, you have a scenario where pH matters, because you can actually get enrichment of the nitrogen pool through the volatile loss of ammonia. And so some of these points up here are in, whoops, sorry, well, can I go back? So Last Chance Lake is actually much higher pH than the other ones, and we see these enrichments most strongly within that system. Okay, so total lipids, how much lipid is in these phases? And so these I'm showing you in terms of milligrams per liter filtered for the fluid phases, or per gram solid within things like the sediments and the salts and whatnot. And so if we look at this, you can see a decrease in abundance when you go from the water to the brine to the ice within our lipids per liter. And so if the brine was just concentrating cells, we would expect the opposite trend, right? Because it's going to sort of push the cells into the brine, and then there's going to be less in the ice, more in the brine, and so on. And so you do see some degradation. And then if we look at the sediment phases, sorry, I keep getting the laser, they're both green. If we look at the sediments, you can see that the sediments have essentially the same amount of organic matter as things like the microbial mat, and they're nearing brine shrimp. And so these sediments are really organic rich, and they have a lot of extractable lipid in there, which is suggesting to me that we've got some really preferential preservation going on within these sediments, consistent with it being a salty, environment. You can also note that the abundance varies a little bit by lake. So you see the light blue ones are down here in Last Chance Lake, whereas those Basque lakes seem to be preserving more extractable lipid. And so we're interested in sort of delving into those trends, see how they compare to chemistry, and sort of flesh out some of these hypotheses we went in with. So how much time do I have? 2 minutes? Great. So this is hot off the presses data, which is why it's in Excel and not something prettier. So this is showing you those hydrolyzed lipids, just a subset that have been quantified for their fatty acid characterization. No. Okay. So if we look at these in a little bit more detail, we see that the waters are a little bit different from the ices, a little bit different from the sediments and mats. In particular, I think it's interesting, these sediments seem to be incorporating some long-chain fatty acids, or sort of mid-chain, that are more characteristic of algal primary productivity when you get into the paleoclimate literature. But you do see sort of strange peaks. So this is a branched fatty acid showing up in the ice that's not really in anything else. So we definitely see some differences, and this is just a subset, so we'll delve into that in the future. And so then the next steps, this is my incoming graduate student, Floyd, and so he's going to take this project over from departing undergrad, and we're going to actually look at those intact polar lipids. So this is what I showed you before. This is just the fatty acid skeleton, but lipids actually come in big boy molecules like this, and there's a lot of structural variability that is interesting, and you can learn a lot more about who's making what based on the intact polar lipids. So, and so life markers, usually they're thought to degrade very rapidly, but we don't know how that works in salt. So we're going to figure it out. So we're going to do the modern sample set and then see how that extends in time, both through some controlled experiments and then through an extension into the sedimentary archives, which Alex will tell you a little bit more about. The bottom of these cores are actually quite old, and so we'll be able to track these biosignatures as they go, or as they are accreted through time to really understand what degradation looks like. And with that, I will thank you for your attention, NASA for money, and all the people that helped make this possible.
All right, we have time for a couple of questions.
It's a race.
Hey, so can you talk a little bit more about the specific fatty acid that you saw in the ice and like why that might be there or what exactly that is?
I have no idea. So literally Hannah sent me that data like 3 days ago. And so I blotted it up. So branched fatty acids tend to be really characteristic. So they're only made by bacteria. You don't get eukaryotes making those. So that's definitely not an algal marker. Why it's in the ice and not in the fluids, I don't know. But that was not all of the fluids. And so what I haven't done is compared like that ice to its paired fluid to its brine to see, if it's like one of those brine pools in Basque Lake is making some weird branched fatty acids, or if it's something else. So it's a rich data set that's only going to get richer as we get all of the samples. Unfortunately, for any GCMS people out there, these samples are really hard on GC columns. Like they're just destroying the columns. So I'm trying to figure out what is in there that's doing that. I think it's organic sulfur molecules, in addition to maybe some other stuff. So, suggestions. So it's a difficult data set now to generate, but we're going to wade through it.
Those are really cool systems. My question is kind of related. You showed some decrease in concentration. Is there any extraction efficiency challenges with some of these salts?
It's a good question, and I don't know the answer. The extraction buffers themselves are pretty salty, and so I wouldn't predict so. So the Bly-Dyer process we do via pretty intense sonication in a single-phase liquid that then separates into a two-phase organic extraction. And so so these each got sonicated. It's like 5 rounds sonicated for 10 minutes, which should be pretty disruptive to lipid membranes, but it's certainly worth testing. Yeah.